Hello, I'm Aarushi, and this is my presentation for WI38 cell lines. So before we get into it, we should talk about cell division. This is how our cells grow, replicate, and divide to create new identical cells. Let's start with interphase. Interphase is the longest part of the cell cycle. It's when the cell isn't dividing, but it's preparing to divide. During this time, the cell grows, performs its normal functions, and duplicates its DNA so that when division happens, each new cell will have a complete set of genetic information. Interphase also has three stages, G1, S, and G2, which is just here. In G1, the cell grows and makes more organelles and proteins. In the S phase, the cell copies its DNA. Then in G2, the cell continues to grow and gets ready for mitosis by making the structures it'll need to divide. Once the interphase is complete, the cell enters mitosis. Mitosis has five main stages: prophase, prometaphase, metaphase, anaphase, and telophase. And well, cytokinesis, but it's not in the picture. In prophase, the cell DNA coils up tightly into divisible chromosomes, and the spindle fibers begin to form. During prometaphase, the nuclear envelope breaks down, and the spindle fibers attach to the chromosomes. Next comes the metaphase, where all the chromosomes line up neatly in the center of the cell. This makes sure each new cell will get an equal set of chromosomes. Then in anaphase, the sister chromatids, which are identical copies of each chromosome, are pulled apart to opposite sides of the cell. Finally, in telophase, new nuclear membranes form around each set of chromosomes, and the DNA starts to uncoil again. So once mitosis ends, the cell isn't quite done. It still needs to divide the rest of the cell. This final step is called cytokinesis, where the cytoplasm splits, forming two new identical daughter cells. So what is the WI38 cell line? The WI38 cell lines were isolated from the lung tissue of a 3-month-old female fetus. It's not an embryo, a fetus. So let's talk about the morphology. It's a fibroblast. The tissue is from a lung. Disease, it has no diseases. And it is initially taken for doing research in 3D cell culture and bioproduction. And to keep it frozen, it requires liquid nitrogen. The introduction of WI38 provided a way to grow viruses in a controlled, reproducible, contamination-free human cellular environment. Because WI38 cells were derived from a single carefully screened fetal donor, so it did not have any prior viruses or diseases. It was sterile and free of latent viruses commonly found in animal tissues. It is genetically stable over many cell divisions. However, later on we'll talk about the half-life limit, which isn't really true. Well, after half-life researches, he figures it out. Capable of supporting the growth of medically relevant human viruses, they became a gold standard in vaccine science. So researchers, what they do is like they test it as a vaccination against many human viruses. And why is this important? WI38 cell line is the first human diploid cell line to be used in human vaccination preparation. As the first human diploid cell line approved for vaccine production, it has been instrumental in biomedical research and biomanufacturing. Its susceptibility to a wide range of human viruses, coupled with its genetic stability, has made it instrumental in the production of vaccines against diseases such as rubella, rabies, and adenovirus, the pictures of which are shown here. So this is rubella, this is rabies, and this is adenovirus. So these are all viruses for which WI38 cell line has provided vaccination against. So the medical impact. WI38 cell lines provide a non-cancerous, stable, and contamination-free environment for growing viruses used in vaccines. This cell line was critical for the development of vaccines against rubella, German measles, rabies, adenovirus, varicella, chickenpox, polio, and hepatitis A. So, for example, let's zone in on rubella vaccination and how WI38 impacted it medically. So, before its introduction in 1969, rubella epidemics caused thousands of miscarriages and congenital birth defects worldwide. The vaccine produced using WI38 has since prevented millions of cases of congenital rubella syndrome. According to the studies from the National Institutes of Health, billions of vaccine doses produced with WI38 has been administered globally, significantly reducing childhood mortality and morbidity from viral diseases. So let's talk about the history a little bit. The WI38 cell line was established in 1962 by Dr. Leonard Havlik and Paul Moorhead at the Wistar Institute in Philadelphia. The cells were derived from the lung tissue of a 3-month gestation female fetus that had been selectively aborted in Sweden. Havlik demonstrated that normal human cells could divide only a limited number of times before senescence, a finding that became known as the Havlik limit. So the Havlik limit, and there's a picture right here. The Havlik limit basically says that since we learned about telomerases in MCB 150 before, and that basically prevents the shortening of telomeres of chromosomes. But sometimes it's inevitable that there will be shortening of telomeres of chromosomes. So once a cell undergoes cell division and then undergoes it again multiple times through multiple generations, the telomeres shorten, and after a point, it's no longer the cell is no longer useful after a certain point. So that's what Havlik has demonstrated, that normal human cells could divide only a limited number of times before senescence, which is basically cell death. And this whole topic is Havlik limit. Havlik limit. So, some ethical concerns. The use of fetal tissue sparked ethical debates that continue today. Critics argue that using cells derived from an abortion raises moral concerns, while others contend that the tissue obtained with consent and used decades ago has saved countless lives without promoting future abortions. So, you're basically saying that you're using one person's cells from an abortion to prevent multiple future abortions. So it's one versus many. Sacrificing one versus gaining for many people. These debates led to the development of stricter ethical guidelines, including informed consent requirements and oversight by institutional review boards, IRBs. Another thing that I didn't mention here is that Havlik, who was a person here, Dr. Leonard Havlik, he gained a lot monetarily because of his research here. So he did a lot of research and then he sold WI38 cell lines to other institutions for more research, and that's where he gained money. But the parents, the mother who had this like abortion child, did not gain anything in value or like monetary wise. That's why people were also angry with Hayflick because they thought that Hayflick was profiting a lot using someone else's cells. So influencing public health. WI38 cell success has also influenced public health policy, demonstrating that human cell strains could replace animal-based systems that were prone to contamination. So before, researchers used to use like monkey kidney cells to test out different vaccines, but now they could use WI38 cells, which are basically human cells, to try out different vaccinations. The shift set regulatory standards for vaccine production and biosafety practices that are still in use today. For example, the introduction of the rubella vaccine produced using WI38 in the late 1960s directly led to widespread rubella elimination campaigns across North America and Europe. After the vaccine came out for rubella, they held elimination campaigns everywhere to prevent rubella from spreading and providing vaccination to everybody. So a little bit of a personal reflection is that this WI38 cell line's legacy deeply affects my own life and the lives of those around me. Many of the vaccines we receive as children, including MMR, chickenpox, and hepatitis A, trace back to this research. Because of these vaccines, diseases that once caused widespread death and disability have become rare in my community. So I'm definitely grateful for that. Ethically, I believe that the use of human-derived cell lines can be justified when the tissue is obtained ethically and when the resulting science serves the greater good. The case of the WI38 demonstrates how one medical discovery handled responsibly can protect entire generations from preventing diseases. It underscores the importance of balancing scientific progress with respect for human life and ethical integrity. These are my citations. I used an article from Nature, and these articles are from Google. And that's the end of my presentation. Thank you for listening.